discuss a topic regarding the apoptosis, which is a common topic in pathophysiology. So, if you are studying in the last minute for the exam for any kind of students, MBBS, pharmacy, nursing, any healthcare professionals, so this is a topic which is very important. So, you can refer the textbook Harshmohan and Robinson book. So, I will just give a brief idea regarding the apoptosis. So, apoptosis means it's just nothing. It's a internally uncoordinated cell death. Means cell is going to die. Cell is going for suicide. So, another name for apoptosis is suicide. Basically, cell is going for suicide. So, we have taken this term from the Greek word called falling off. The meaning is falling off. Basically, the cell is going to suicide. So, before that, I will just give a brief introduction regarding how the cell can maintain everything when the cell will go for suicide. So basically we can consider this is a cell, isn't it? You can see this is a cell. So basically if anything happens for us, like I'm doing some excessive exercise or any injury, cell will try to maintain that stability. That which is called homeostasis. So cell will try to maintain that stability. Again, if the cell is getting any stress, cell is going to get what? Stress. Severe stress or what? Then cell will go for reversible cell injury. Reversible cell injury. Then again, the cell is not going for, like the cell is getting so much of stress, then again cell will go for irreversible cell injury. So once again understand, if anything happens to our cell in our body, cell will initially try to acclimatize. Cell will try to steady that particular agent. So that is the process called homeostasis. Again, if the cell is getting so much of stress or any injury, it can be anything, any etiological factor. Etiological factor means any causative agent. Then cell will go for reversible cell injury. In this time, what the cell will do? Cell will go for some adaptation, which is called as cellular adaptations. Cellular adaptations. It can be hypertrophy, atrophy, hyperplasia, etc. So trophy means understand, that means size. Plasia means understand, it is number. So hypertrophy means size is becoming more. Hyperplasia means cell is becoming not size. Cell number is increasing. So, when the cell will go for cellular adaptation, the cell is getting irreversible cell injury. But if the cell is going for irreversible cell injury, cell will not go for any type of adaptation. Cell will go directly for death. Cell death. So, what is the cell death? If the cell is getting an irreversible cell injury, we can't reverse that situation of the cell back to the normal state. Here, the cell can go back to the normal state. Here, cell can go back to the normal state, but here the cell won't be able to go back to the normal state, which is cell death. Cell death, different types are there, okay? Cell death, different types are there. So, first type is autolysis. Autolysis, second necrosis, and third one apoptosis. So, autolysis, necrosis and apoptosis. So, autolysis means nothing, auto means self. The cell is going to digest. Self-digestion. Autolysis means self-digestion. Necrosis is also cell death. But the thing which differentiates necrosis and apoptosis is, here you can see inflammation. You can see inflammation in the cell. But in the apoptosis, there will be no inflammation. Inflammation will not be there. Okay. In necrosis, inflammation will be there. In apoptosis, inflammation will not be there. So that is the main difference which segregates this necrosis and apoptosis. Okay, clear? Then again, apoptosis, it can be because of some physiological or pathological. Okay, physiological or pathological. But necrosis, it will be causing, the causative agent will be something, any injury, any toxins, any free radicals, it can be anything. Okay. The thing which you have to remember is inflammation will not be there in apoptosis, but inflammation will be there in necrosis. How you can remind? You can remind it with NN. Okay, N. Here inflammation N is there, necrosis N is there. So that means inflammation will be phagocytosis, which is a common step in both necrosis and...
So before we move to the introduction of apoptosis, I will just briefly just say regarding the three morphology of irreversible cell injury. Three morphology of irreversible cell injury. The first one is autolysis. Autolysis means auto means self. Self, the cell is digesting itself. Okay. Then second one, necrosis. Necrosis means cell death, where the cell will swell or the cell will burst. There will be inflammation will be there, because how you can remind is N, there is N for necrosis and N there is for inflammation. And in apoptosis, the cell will shrink. The cell will shrink and inflammation will not be there. This is also cell death, where suicide will happen, because it is a programmed cell death. Everything is programmed. The cell will go for suicide. So the definition is coordinated and internally programmed cell death. You can get the definitions from every book. In every book there is there, like the Harsh Mohan book and the Robinson book. According to the comfortable level, you can take the definition. And apoptosis, we have taken this term from the Greek word, means falling off or dropping off, like the leaves are dropping off or falling off. Like that you can just remind it. Then the next thing is, there will be no inflammation in apoptosis. I already told you, which is a very distinctive feature that we can segregate in apoptosis and necrosis. Inflammation will not be there in apoptosis. Why? I told you one reminder, N is there in necrosis, N is there in only inflammation. So apoptosis, N is not there, so there is no inflammation. Why inflammation is not there? Because after the digestion, after the fragmentation, the disintegration, the contents of the cell, cellular contents, the contents of the cell, it is not going to get released. It is not going to get released into the fluid. Okay, so that's why once it is released into the fluid, only it will show the characteristics of inflammation, the 5 characteristics. So there will be no inflammation. Why? Because the contents of the cell is not going to get released into the body fluid. Again, one more thing, distinctive feature is the cell will shrink. The size gets reduced because of the dissolution of the nucleus, the condensation of the nucleus, the fragmentation of all the cytoskeletal proteins, nuclear proteins, and all disruption of the reticulum, organelles, everything, and the loss of the integrity of the cell membrane. So as a result, the size get reduced. As a result of the reduction of the size, the cell will shrink. But in necrosis, the cell will burst or the cell will swell. Then we can just find out which cell, why the cell is going for a suicide. What is the reason that cell will go for suicide? The first reason is, once if the cell is not getting any growth factors or any hormones or any signal in the sense like not getting sufficient growth factor or hormones at that time, or the cell is feeling like the function is over, so at that time the cell will go for apoptosis. The second thing is, if the DNA of the cell or the nucleus or the protein is damaged, and it is beyond repair, you can't repair that DNA, at that moment also the cell will go for apoptosis. So these are the two reasons that the cell is going for apoptosis. The cell is going for a suicide thought. One is if the cell will feel like the function is over, or it is not getting deprived, it's not getting any growth factors or hormones, cell will go for suicide. Then again, the thing is, if the DNA of the cell is...
So we have already completed the apoptosis introduction and all. So that I have taken from the Harsh Mohan pathophysiology book. So now we will see what are the causes. I told you in the before, there are causes for the apoptosis. It can be physiological or it can be pathological. Okay. So physiological means what? What is the difference between physiological and pathological? Pathological means you have to understand it is because the suicide. Apoptosis means what? Suicide basically. It is because the cell is going for suicide because of some pathological condition. Pathological means what? Some disease condition. The cell is going for suicide because of some disease condition. Here, the cell is going for suicide, right? Apoptosis means what? Suicide. The cell is going for suicide because of the physiological condition. Physiological condition means the condition which happens normally in our body. These are not because of any disease or what, which happens normally in our body. That is commonly it is happening in our human body. So in that, first we will learn regarding physiological apoptosis. Okay. So what I said, physiological apoptosis means cell is going for suicide. That means cell death will happen. Obviously cell death will happen. Apoptosis means obviously death will happen because of some normal condition, not because of any disease or not because of any chemical reaction or injury, any free radicals, toxins, no. Because of the normal condition of our body. So for that we can take an example of embryogenesis, which is already in your Hirschman textbook. You know, we all were embryos, right? We all were, suppose this was a uterus, the womb of a woman, of a pregnant woman. And we all were fetus, right? We all were small fetus. At that time, how our hands, legs were, how our hands were, whether our hands were like this, whether it was like this, no, right? Whether our feet was like this, no, right? So how it was like this? It was covered, right? It was covered like this. It was like this. Well, we were in the fetus. Once the baby is born, how it has become? Suppose if it is leg or suppose it is hands or something. Once the baby is born, how our hands? It is very distinctive, right? Yes, our hands is like we have 5 fingers, we have 5 toes and all, right? It is very distinctive. So what happened? What happened in this process? There were cells in between, right? There were cells in between your digits and there were cells in between your toes, right? So that cell went for suicide. That cell has gone for suicide. The cell undergone apoptosis. So we got this distinctive digits and toes. So here cell is going for suicide. Cell is dying in between these digits and toes because under a normal condition, right? That's why it is called as physiological apoptosis. Why it is called as physiological apoptosis? It is a normal condition, right? Why normal condition? Pregnancy. Pregnancy is a normal condition, right? Pregnancy is not a disease. Pregnancy is not a disease. That's why this procedure comes under the physiological apoptosis. I hope you are clear. So it's nothing. During the embryogenesis, the cells in between your toes, in between digits undergo apoptosis. That is the process, the physiological apoptosis. Because pregnancy is a physiological process, because pregnancy is not a disease. Again, we can take one more example, simple example. Okay, so once you are pregnant, after the parturition means after you deliver the baby. The parturition is a medical term. After you deliver the baby in your uterus, from the uterus will undergo hypertrophy and hyperplasia, right? During the pregnancy condition, obviously the womb, the stomach will increase, right? I mean, it will become big, right? How? What is the process happening there? Hypertrophy and hyperplasia means the cell number will also get increased, cell size also get increased. Once you deliver the baby, after the parturition, again the cell will come back to the normal place. The womb, the stomach, it will come back to the normal place. How? Apoptosis happens. The cell will undergo after the parturition. The cell again, the woman will come back to the normal state. How? That hypertrophy, hyperplasia which occurs in pregnancy. After the parturition, again it will come back to the normal place. How? That cells, all the excessive cells which undergo apoptosis. Which apoptosis? Physiological apoptosis. Why physiological apoptosis? Because it is a normal procedure. Pregnancy, parturition, which all are a normal procedure in our body. It's not because of any disease or not because of any pathogen or any organism. Understood? So these two examples we can learn for an easy thing, which is very, very easy. Okay. Next, we will go for the pathological apoptosis. Pathological apoptosis means what? Because of some pathogen or because of some organism, because of some agent, apoptosis is happening. What? Cell is happening. So some disease should be there, right? So the common disease in your Hirschman is AIDS. Okay? AIDS is acute immune deficiency syndrome, which is caused by which virus? HIV. HIV and AIDS both are not same. HIV is a virus which will cause AIDS. AIDS is like the last stage of a patient who is having an HIV. Okay? So AIDS. In the AIDS patient, what will happen? Do you know? I will not go detail into that. Do you know whether we will check the CD4 count? CD4 count of a patient. CD means cluster of differentiation. The cluster of differentiation, the CD4 count we will count in a patient who is having infected with HIV, because usually the normal level will be like 500 to 1500 cells per mm cube. So if you are infected with HIV, if you are infected with AIDS, the last stage your CD4 count will become less. Why? It is an immune cell, because HIV is basically affecting our immunity. So once it affects our immunity, which cell is going to get affected by this HIV virus, this AIDS stage, this disease? So lymphocytes. Which cell? Lymphocytes. What is a lymphocytes? It is a type of WBC. It is a type of WBC. So WBC we have 5 types. WBC we have 5 types. You know that: lymphocytes, monocytes, neutrophils, eosinophils, etc. So out of that 5 types of WBC, it is going to affect lymphocytes. It is going to affect the lymphocytes. So out of the lymphocytes, how many types of lymphocytes are there? How many types? B lymphocytes, T lymphocytes. Okay, clear? B lymphocytes, T lymphocytes. Again T lymphocytes. Whether we can classify T lymphocytes? Yes, we can classify T lymphocytes into CD4 cells, into CD8 cells, memory T cells, natural killer cells, T cells, many things. Okay. Again we can classify the T lymphocytes into CD4 cell. CD4 cell, see, CD4 cell. CD4 cell, another name for CD4 cell is helper T cell. Helper T cell. CD8 cell, another name is cytotoxic cell. Then again memory cell is there, then natural killer cells are there, many cells are there, but we have to just remember CD4 count gets affected. How it is going to get affected? It will decrease, the value will get decreased. So when the HIV affects our body, which lymphocyte will get affected? B or T? T get affected. From the T, it is going to get affected? CD8, memory T cell, natural killer? No, CD4. How you can memorize? Because obviously CD4 count will become less in the patient who got affected with the AIDS. I hope you are clear. Then again, if you see for the chemotherapy case, second we can take a simple example, chemotherapy case. Chemotherapy means what? The therapeutic agents we will give for killing cancer. Nowadays we can the word killing cancer cell. So suppose you are having a cancer. Obviously we have to take a chemotherapy, right? Other than the surgery, radiation and all. There are various options are there, but when you give a chemotherapeutic agents, the cancer cell will get destroyed, right? Cancer cells, how? It is going apoptosis. The cell is going to die, right? It is undergoing apoptosis. What apoptosis? Pathological apoptosis. Why pathological apoptosis? It is because of a disease, right? The disease here is cancer, and we are giving chemotherapeutic agents, and cell is going to die. Why we are giving therapy? To kill the cell, right? So what cell is going to get killed? Cancer cell. Okay, I hope you are clear.
The types how apoptosis can happen. So basically it is divided into 2, physiological and pathological. So physiological means the cell will go for suicide under a normal condition like pregnancy, after the parturition or during the embryogenesis, because there is no pathological state, right? Because pregnancy is not a disease, right? So the death will happen. Whose death? Cell death will happen. How? By apoptosis. At one condition, physiological condition. Physiological means under the normal condition, the body is feeling like you don't need this much of cell, right? Because after the parturition, there will be a decrease of a cell, right? Cell number and the size. Because you don't need that much amount of a cell or cell size after parturition, right? But during pregnancy, we need hypertrophy and hyperplasia. Why? To accommodate the fetus, right? So once the parturition, after delivering the baby, we don't need that cell or the cell size or number. So our body feel like these cells are not needed for the body. So the cell will go for suicide. So which is very physiological process, which is a very normal thing, not because of any disease or injury. So example you can see during the embryogenesis, like during the organogenesis, during the organ formation inside the womb, you can see before the apoptosis, you can see like there were cells in between the fingers and the toes of the baby. But once you get the organogenesis, the organs are formed distinctively, very distinctively. The cells in between all these digits, these fingers, toes and all, it will undergo cell death. It will undergo cell death. Then again, second thing, I hope first example is clear. It's just nothing. Just keep in your mind that once the formation of organ very distinctively from that very small fetus, obviously we need distinct organs, right? So some cell has to undergo death. Okay. Then second example, during the lactation, the pregnant women breast will be it will be very big, right? So the cell has to, after the weaning means after the stoppaging of the lactation for the baby, so after that regression of will be there. Regression, like the cell will undergo death because many cell will have died after the weaning of breast. Why? Because during the lactation time it will be there, right? The cell will be more. So after you stop giving milk and the lactating milk to the baby, the cell will undergo death. This is a very normal phenomena. It is a normal phenomena. So that's why these all examples comes under physiological apoptosis, not pathological, because all these conditions were not a disease. Okay. Then again, when you go to the pathological apoptosis, means it is the cell is undergoing or cell is going for suicide because of some disease that has happened to our body. Because of some disease happened to the body. So what are the diseases? I have already taken the example. AIDS, HIV. So HIV virus will affect the body. It will cause AIDS. The immune deficiency will happen in the body. So eventually it will affect the immune cells in our body. In that immune cells, the T cells, CD4 cell get affected by this virus. So eventually the depletion and the death of the CD4 cell in our body will happen. The death of CD4 cell in our body will happen due to this virus, which is a pathological apoptosis. Why? Because a pathological cause is there. What is the cause? AIDS, HIV virus. Again, you can see in this tumor cases, chemo cases, in the cancer cases, again cancer is a pathological state. At that moment also, when you give a chemo drugs to kill the cancer cell, obviously the cell is undergoing death, right? So because of the pathology, what is the pathology here? Cancer disease is there. So that's why it is coming again under the pathological apoptosis, because cancer is not a normal phenomenon. It's not a physiological phenomenon.
So before we go to the pathway, we'll just rewind it, we'll just brush things. So for the causes of apoptosis, it can be physiological or pathological. I hope you're clear, the definition is clear, I guess so. Then the pathways are there. For how the apoptosis is going to happen, the mechanism, how it is going to happen. There are two mechanisms are there, two pathways are there. One is extrinsic and another one is intrinsic. Extrinsic, another name is cell death receptor pathway, pathway, and intrinsic pathway, this another name is mitochondrial pathway. Again it is divided into two. Again it is divided into initiation and execution phase. Again here also initiation phase and execution phase. So before coming to that, I will just give a brief introduction regarding this. All our cells, when I will be having a caspases enzyme. Caspases means it is a group of enzymes, it's a group of proteolytic enzymes. C for cysteine protease, ASP for aspartic acid. When ACE, obviously we know why we are giving ACE for enzyme, right? So caspases means it contains a lot of enzymes, 8, 9, 7, 3, a lot of enzymes are there in this group of enzymes. So we all have this caspases enzyme, okay? Everyone will be having caspases enzyme. Once the caspase gets activated, the cell will go for suicide. The cell will die. Then why we are not dying? We also have this caspase enzyme. Why we are not dying? Because the caspase is in an inactive form. Once the caspase gets activated, cell will go for suicide. So during the apoptosis, what happens? Caspases will get activated. Caspases will activate, it will disintegrate and it will form apoptotic bodies. Apoptotic bodies means the bodies just undergo apoptosis. So in this apoptotic bodies, it contains a small amount of nucleus, small amount of cell membrane, small amount of organelles from the main cell. From the main cell, you can see apoptotic bodies. Okay, when the cascades will get activated because of some stimulation. Some stimulation will happen during the extrinsic or intrinsic pathway. When the cell will go for suicide, when will the cell go for suicide? When if they feel like the DNA is damaged, DNA is already beyond the repair, then it will go for suicide. Again the cell will go for suicide. At what time? If you feel like the function of the cell is already over, the function of the cell is over, and the cell will go for suicide, right? As people will go for suicide likewise. Okay, so at that time we will send some signal through this pathway, and finally what happens, the caspase, the inactivated caspase will get activated. As a result, apoptotic bodies will be formed. How apoptotic bodies form? Small amount of nucleus, small amount of organelles, small amount of cytoplasm, everything will be there. And what happened after these apoptotic bodies? These are apoptotic bodies, right? Which undergoes, who will come for phagocytosis? Who is doing that phagocytosis? Macrophages. Macrophages will come for phagocytosis, and it will end up all the apoptotic bodies. I hope you understood. One more thing, phagocytosis will happen both in apoptosis and necrosis. And again, one more differentiation I will tell. In apoptosis cases, cell will shrink. In necrosis cases, cell will swell. Cell bursting will happen in necrosis cases. In apoptosis, cell will shrink. Again, one more I told, inflammation will happen in where? Necrosis, not in apoptosis. Okay.
So again, the pathways of apoptosis, it is divided into extrinsic pathway and intrinsic pathway. Extrinsic pathway means the cell is getting signal from the external environment, from the outside to the inside. So it is divided into two phases, initiation phases and execution phase. Then in the intrinsic pathway, the cell is getting a signal. What signal? Apoptotic signal. Why they are getting apoptotic signal? It is giving a signal to the cell to undergo apoptosis. So in the intrinsic pathway also there is two phases, initiation phases and execution phases. Okay. So before we move to the pathway, I just want to give a brief thing regarding the mechanism. The another name for intrinsic pathway is mitochondrial pathway. Another name for extrinsic pathway, cell death receptor pathway. Why it is mitochondrial pathway? The intrinsic pathway is called as, that I will explain later. But because mitochondrial permeability is involved, that's why the main thing is coming from the mitochondria. That's why we have given the name mitochondrial pathway. Extrinsic pathway, it is known as death receptor pathway, because that pathway, it is initiated. That pathway starts from the death receptor, because death receptor plays an important role in that pathway. That's why it is called as death receptor pathway. Okay. Then again, caspases, I have already told you regarding the caspases. A cell in all our body, caspases will be there. It is like a group of proteolytic enzyme. It contains a lot of types of enzyme, 3, 7, 6, 8, 9, many enzymes are there, almost 10 enzymes are there. So it is derived from the term, I hope you know this thing, caspase, C4 cysteine protease, ASP for aspartic acid, AAC form. Obviously we will term an enzyme with A's, right?
Regarding the apoptosis introduction and what are the causative agents for the apoptosis, we already discussed physiological and pathological, and now we are discussing regarding the pathways, right? How the mechanism, how the apoptosis will happen. So I told you extrinsic and intrinsic is there, and intrinsic initiation and execution phase is there, and extrinsic also initiation and execution phase is there. So first we will study regarding the extrinsic pathway. Why it is called as extrinsic pathway? Because we are getting the apoptotic signal. Apoptotic signal means what? The signal which will induce apoptosis. Apoptosis means what? Suicide. So we are getting extrinsic pathway. Extrinsic pathway means we are getting signal for what? Signal for what? Apoptosis. So we are getting the apoptotic signal from the outside. So we are getting the signal from the outside. So that's why it is called as extrinsic pathway. I hope you are clear. So in the extrinsic pathway, it is initiated through the death receptor. So receptors in our body. In every cell, death receptor will be there. So these are death receptors. See, these are death receptors. Death receptors is divided into two. One is TNF, and another one is FAS or CD95. Don't get confused with that CD4, CD8 and all. This is CD95. Okay. So once again, extrinsic pathway is also divided into two: initiation phase and execution phase. Why it is called as extrinsic pathway? Why it is called? Because we are getting the signal from the outside. What signal? The signal for apoptosis. The cell has to go for suicide. So that is mediated through some death receptor. What are the death receptors? TNF also already you know, tumor necrosis factor, and FAS, or the other name is CD95. So I hope you can see CD95. See, you can see a lot of CD95 or FAS protein present receptors present on the cell. So how we can initiate the apoptosis? We have to initiate the apoptosis, right? So when the ligand comes, what do you mean by ligand? Signal only. What signal we are getting? The signal for apoptosis. So we are getting that signal, that apoptotic signal in the form of a ligand. So suppose the death receptor is CD95. Let it be TNF? No, we will not take TNF right now. So we will take CD95. Other name for CD95? FAS. So if you want to receive the ligand, what do you mean by ligand? The apoptotic signal which is coming in the form of ligand. So if you are taking CD95 or FAS, which ligand will come? Obviously FAS ligand will come. Suppose if you are going to take the TNF receptor, here if it is TNF, means which ligand will come? FAS? No, TNF ligand will come. Okay, don't get confused. For CD95 or FAS, which ligand will come? FAS ligand only will come. So now you can see the spacing between the receptors, right? We have some space, right, in between the receptors. So once you get the signal, what signal? Apoptotic signal. How signal is coming? In the form of FAS ligand. Once you get the signal from the outside, why outside? Because it is extrinsic pathway. Once you get the signal from the outside, see all these death receptors now, it will come closer. See, it will come closer together. And that area, that is called death domain. That is called death domain. Since FAS ligand is coming and binding with which receptor? FAS receptor. Other name is CD95. Why it is coming together? Because the ligand has come. So it will come together and it will form a death domain. So here it is FAS. So that's why it is called as FADD. Fast associated death domain. Fast associated death domain. How domain is formed? Because the ligand came. Why it is FADD, not TAR, ADD and all? Because the ligand here is FAS. That's why. So once the ligand comes, it will form a death domain. Once the death domain is formed, death domain means it is the triggering point. It is the triggering point. So once the death domain is formed, right? I told you domain, that death domain. Why it is called death initiator? Death initiation. The domain. We are initiating the death. We are initiating what death? Suicide, cell death. Okay. So once it is formed, it will activate the procaspase 8. Procaspase 8. I told you already. All our cells having caspases, a group of enzymes. I told you a lot of enzymes are there like 8, 9, 7, 3, etc. etc. So we have to take 8. Okay. Don't forget extrinsic pathway. And once again telling extrinsic pathway. What phase we are saying? Initiation phase. Initiation phase. So basically procaspase 8. Which caspase it is? It is a initiator caspase. It is a initiator caspase. Why it is initiator caspase? We are talking bodies. So one thing you have to remember is, in the extrinsic pathway, initiation phase, procaspase will be there. And one more thing, you might have a doubt, why initiation phase is there, why execution phase is there for every intrinsic pathway and extrinsic pathway? Because in the initiation phase, what happened? So the initiation phase, what happened when initiation happened? What initiation? Initiation of the caspases. So that's why we have an initiation phase in the extrinsic as well as intrinsic pathway. So in the initiation phase, what happens? Initiation, activation of the caspase. See, whether activated? Yes, caspase was activated, right? Which caspase was activated? 8. Caspase number 8 got activated. And in the execution phase, what happens? Execution happens. Why? Why execution is happening? See, once the caspase got activated, it will go for execution. The cell will go for suicide. So that's why we have two phases in each pathway. Initiation and execution phases, both pathway. Why? Because initiation, that activation of caspase will be there, and in the execution phase, once the caspase got activated, we are sure, we are 100%, 7% sure the cell will go for apoptosis. It will form apoptotic bodies, then phagocytes will come, which will enter the apoptotic body.
So when you see the extrinsic pathway, I have already discussed, you can see, right? See? Ligand. You can see the ligand. FAS ligand. FAS another name is CD95. CD95. Okay? CD95. So you can see the ligand is coming, right? FAS ligand. I have already told you it can be FAS ligand, CD595 ligand, or it can be TNF ligand also. So according to your wish, you can change FASL or like TNFL. Okay. So again, you can see the death domains. You can see the death domain. Yeah. See, before the death domain was like this. Okay. Space was there between before the coming of ligand. Okay. Once the ligand comes, see, every death domain came close together. See, they came close together. Why? Because see, the ligand came, that's why they came close. So they are forming a death domain. See, FAS ligand, it came, and that domain from every space it came together and it formed FADD, fast associated death domain. Instead, it will do, this death domain, it was a triggering point. Okay, it was a very triggering point to activate the caspase. Why we are activating caspase? Because caspase enzyme is the one who helps for the apoptosis, right? In our body, caspase is in inactive state. So FADD, this will activate the caspase. So eventually, if caspase gets activated, obviously cell will undergo death. So here, which death domain is formed? FADD. It eventually activate procaspase. It was an inactive caspase. Inactive caspase got activated to caspase 8. Only thing you have to remember is number 8 for extrinsic pathway. So as a result, this activated caspase, it will activate the executioner caspases like 3, 6, 7. And as a result of the executioner caspases, the cell will undergo cytoskeletal damage, nuclear fragmentation, everything. The cell will undergo. The cell will shrink. Then again, the cell will form an apoptotic bodies. See? So if this was the cell, the cell will form an apoptotic bodies. This apoptotic bodies contain small amount of nucleus, small amount of cytoplasm, small amount of organelles, and see, apoptotic bodies are formed. And after that, this apoptotic bodies are engulfed by phagocytes. Okay, this apoptotic bodies undergo phagocytosis by the phagocytes. I hope it is clear. Only thing you have to remind is, once the ligand comes, it can be FASL or a TNF. The death domain will come closer together. It will form a death receptor. It can be CD95 or a TNF or a TNF, this will come closer together and will form a domain. As a result of the formation of domain, the caspase gets activated. So this activated caspase 8, it will activate another executioner caspases. Executioner caspases means these are 3, 6, 7, which will execute the apoptosis, okay. So it executes apoptosis. Then the apoptotic bodies will be formed. It is engulfed by the phagocytes. I have mentioned everything regarding the initiation phase, why it is called extrinsic pathway, because you are getting the signal from the outside. And in the initiation phase, you can see 2 types of receptors are there, 2 types of death receptors are there. First one is FAS or CD95. Second one is tumor necrosis factor, TNF receptor. 2 types of receptors are there. One is TNF and another one is CD95. And once the ligand comes, hopefully you can see once the ligand comes, FASL. Yeah, so once the ligand comes and bind to the receptor, see, this is a receptor, okay. This is the receptor. This is the receptor. Okay. This is the receptor. These all are receptors. This is death domain. This is DD. This is death domain. So once the receptor get binded to the ligand, it will form a death domain. And as a result, in the cytoplasm, as the formation of the death domain, it activates the caspase. It will activate the caspase. Which number? Which caspase? 8. And as a result of activation of the caspase number 8, it will activate the executioner caspases like 3, 6, 7. As a result, all these damages will happen. Cytoskeletal damage, disruption of the ER means the endoplasmic reticulum. After that, the apoptotic bodies will be formed. Execution happen, right? What execution? Apoptosis execution will happen. As a result of the apoptotic bodies from the apoptosis, it is again engulfed by the phagocytes. It is engulfed by the phagocytes. Again, how the cell can identify whether it is a phagocytic cell or a apoptotic body? With the help of a phosphatidylserine. So why? Because in a normal cell, this thing will be an inside of a cell. In a normal person, okay? In a normal cell. In a normal cell, it will be inside. But in apoptotic bodies, it will be outside. So that phagocyte can easily recognize which is the apoptotic body. Phagocyte can easily recognize. So how phagocytes can easily recognize to which cell I have to do the apoptosis? Because this phosphatidylserine, it is present outside the cell in apoptotic cases. In normal cases, it is inside. So phagocytes can easily recognize and can do the process, their work, phagocytosis.
So next we will learn regarding intrinsic pathway. So intrinsic pathway, we will get the signal from there. In that name itself it is there, the signal from the inside. Signal we will be getting from the inside, like if the DNA is damaged or something. Obviously, some P53 will come to repair and all. Again, if the P53 is not able to repair and all, obviously that has to go for suicide, right? So signal will come from inside. So that's why intrinsic pathway. Again, in intrinsic pathway also, we have 2 phases. What? One is initiation phase. Next is execution phase. And I hope I have already know why initiation phase in intrinsic pathway. Again, here also initiation, activation of caspase enzyme will happen. Execution, that execution will happen. That apoptosis procedure will happen. Why apoptosis procedure is happening here? Because the caspase has got activated. Caspase has got catalyzed in the initiation phase. Here in the extrinsic pathway, it is activated to receptors, okay, through death receptor, and it is here through the mitochondria. That's why it is called as mitochondrial pathway, because the mitochondrial permeability, the permeability of the mitochondria will initiate the intrinsic pathway. That's why it is also called as mitochondrial pathway. Why happening intrinsic pathway? If you are not getting any sufficient growth factor or any hormones, which is already in your Robinson book, if you are not getting sufficient growth factors, hormones, what happen? Cell will initiate apoptosis. Okay, cell will initiate what apoptosis? If you are not getting any hormone growth factors, it will go for this pathological, physiological apoptosis. Okay. members. We can say these are like members. Anti means it is against apoptosis. It will not allow the cell to undergo suicide. These are members. BCLX, these are like group of oncogenes. These are like BCLX, BCL2, MCL. These are like members which what it will do? It will prevent cytochrome C from mitochondria to cytoplasm. You know, once cytochrome C comes to the cytoplasm, cell will undergo apoptosis. So these are the people, these are the members, these are the people which prevent cytochrome C from mitochondria to cytoplasm. That's why cell is not undergoing apoptosis in our body. So once you are not getting sufficient signal, the withdrawal of growth factor, hormone and all, what happens? This proteins now, this membranes, these people, it is replaced by pro-apoptotic proteins. Pro means you know it will initiate apoptosis. It will be replaced by whole members which people BAK, BAM, BIM, BAX, BAD. Okay, these are the members which promotes apoptosis. So once you get like loss of the growth factors or hormones or something, the signal from the inside. Why from the inside? Intrinsic pathway. Once you not get, what happens? These members, what these members will do? It will prevent cytochrome C from not going to cytoplasm from the mitochondria. So once you get this signal, you are not getting the current signal, when you get the signal from the inside, these members are replaced. These members are replaced by the apoptosis promoting members. What are the members? BAC, BIM, BAX, BAD. These are replaced by these people here. At that time, what happens? Obviously, cytochrome C will come to the cytoplasm. Isn't it? Cytochrome C will come to the cytoplasm. And here, one cytoplasm, here where in the extrinsic pathway, what was the triggering point? What is the triggering point? Death domain, right? FADD. The death domain was a triggering point for that execution phase of apoptosis. So here what is the triggering point? Cytochrome C came. So it has to initiate some cascades, right? Why initiation? Because initiation phase is there in the intrinsic pathway. So it has to initiate some enzyme. What enzyme? There we have discussed caspase 8. So here caspase 9. So 9. So what happens? Once the cytochrome C comes from the mitochondria to cytoplasm, why coming? Because the members of pro-apoptosis, the members who promote apoptosis is replaced by these members are not there, which prevents apoptosis. It is replaced by these members. So obviously the cytochrome C will come from mitochondria to cytoplasm, and it will. One thing you have to remember is pro-caspase was always good with APF. So they were together actually. Apoptosis promoting actuator factor. So these are like together. They were like good partners. Okay, they were good partners. What happens is once cytochrome C comes, what it will do is like it will take this APF. Okay, it will take this APF. So at that time, procaspase 9 will be free. That's what you remember. This APF and procaspase, they are like buddies. They are like good partners. Once cytochrome C comes from the mitochondria to cytoplasm, why coming? Because of the pro-apoptotic members. Once that comes, the cytochrome C and APF will go for some trip. Okay, they will go for some trip because the cytochrome C will take the APF. So who will be remaining? Who will be remaining? Procaspase 9. So again, initiation phase has to occur, right? Initiation phase means what? Activation, the catalyzation of enzyme. So here, the procaspase 9 will convert to caspase 9. It will convert to caspase 9. So initiation phase already occurred. Then again, execution phase means what has to occur? Same procedure. This caspase, once the caspase got activated, it will undergo this disintegration, everything, the fragmentation, this of endoplasmic reticulum, nuclear matrix, everything it has undergo because the caspase got activated. Again, it will form apoptotic bodies. Again, that apoptotic bodies is engulfed from phagocytosis by the phagocytes. Okay, phagocytes.
Pathway you can see. This is a mitochondria. This is a mitochondria. This is like in a bigger picture, in a broader view. This is a mitochondrial inner membrane. This is inner membrane, and this is outer membrane. This is outer membrane. So this is mitochondria in a very broader picture. This is inner membrane and outer membrane, and you can see cytochrome C. See, This is permeability, okay? Like this is permeability. This is showing permeability. This is cytochrome C, which is a phospholipid which is present between the mitochondria and the inner mitochondrial membrane and outer mitochondrial membrane. So cytochrome C, the position of cytochrome C in a normal cell, for example, in our body, the cytochrome C currently is in the position in between the inner membrane and the outer membrane. But once if the cytochrome C comes to the cytoplasm of the cell, okay, their apoptosis starts. It will trigger. That is a triggering point. Once their apoptosis will start. So you can see normally cytochrome C is present in between the membranes of mitochondria. And you will be having a doubt like why this mitochondria, from how the cytochrome C from the mitochondria will come to the cytoplasm and will trigger the apoptosis. Yeah, I have an answer for you. Yes, I have an answer. So there are some proteins, there are some members which helps in the invitation of cytochrome C from the mitochondria to the cytoplasm, and also will prevent cytochrome C from coming from the mitochondria to cytoplasm. So that is like pro-apoptotic proteins. Okay, pro-apoptotic proteins. So I will just show you. The intrinsic pathway is also called as mitochondrial pathway. I hope you are clear right now because of the permeability of the mitochondria is involved. And cytochrome C, I hope you have understood. And so these are the proteins. These are the proteins. You can just note it down. Pro-apoptotic proteins means it promotes apoptosis. It promotes apoptosis. And anti-apoptotic proteins means it prevents apoptosis. How it is preventing apoptosis? How it is preventing? Because it prevents cytochrome C. It prevents cytochrome C from coming from the mitochondria to the cytoplasm, because you know, if the cytochrome C comes from the mitochondria to the cytoplasm, the cell will undergo death. So these anti-apoptotic proteins, what it will do? It will prevent the cytochrome C from coming from the mitochondria to the cytoplasm. So these are the members. These are the persons who actually prevent apoptosis. But once you get all the signal from the inside, then these members are replaced by the pro-apoptotic proteins. Pro means it promotes. So these members will be present. BIM, BAX, BAC, BID and all. Okay. So now we have understood how cytochrome C is coming from the mitochondria to the cytoplasm, because the replacement of anti-apoptotic proteins by pro-apoptotic proteins, the proteins which promotes apoptosis. Okay. So in the initiation phase, you know, I have already talked, this cytochrome C will come. Cytochrome C, it will come from the mitochondria to the cytoplasm, and this procaspase 9. One thing you have to remember here is these apoptosis activating factors. See this APAF. Yeah, once cytochrome C comes to the cytoplasm, it will take this APAF. Okay, see, before APAF and procaspase 9, they were so good. See, they were staying together. APAF1 and procaspase 9, they were staying together. But once cytochrome C comes from the mitochondria to the cytoplasm, see, the cytochrome C is going to take, the cytochrome C is asking APAF to come with me. See, it is going with the cytochrome C. So remnant, who is the procaspase 9. So basically initiator caspase in intrinsic pathways, procaspase 9. So here activation will happen. Again same thing, execution will happen. This caspase 9 will activate 3, 6, 7, and execution means apoptosis. That process will happen, which will eventually result into apoptosis. Apoptotic bodies will be formed. AP bodies will be formed. It will be phagocytosed by the phagocytes. Okay. I'll just tell once again. Cytochrome C, before it was not coming to the cytoplasm because of the anti-apoptotic proteins, which basically prevents apoptosis to happen. So that's why cytochrome C was staying inside the mitochondria. Once it is replaced by the pro-apoptotic proteins, means which promotes apoptosis, cytochrome C comes to the cytoplasm. It will take APAF1 and pro-caspase gets activated to caspase 9. As a result of activation of caspase 9, it will activate other executional caspases like 3, 6 and 7. And what happens eventually? The cell will undergo cell suicide, apoptosis. Okay. As a result, bodies will be formed that will be engulfed by the phagocytes. I hope you are clear. That's what I have written here. I have mentioned the pro-apoptotic proteins name and the apoptotic proteins. Then you can see executional phase. It is similar in both pathway, in intrinsic and extrinsic pathway. It is very similar, and I have mentioned these things also. Initiator caspase is what it will do. It will activate the caspase. It will activate the caspase. It will catalytically active. Then executional caspase, it will execute the apoptosis. It will execute the process. And what are the inducers for the intrinsic pathway? Withdrawal of growth factors or hormones. And what are the causes, inducer for the extrinsic pathway? Interaction between the receptor and ligand. I hope you remember the interaction between the receptor and ligand in extrinsic pathway. See, interaction between the ligand and receptor. See, in extrinsic pathway. Then again, phagocytosis, already we have discussed. It is the dead apoptotic cells, the cells who have undergone apoptosis is phagocytosis by the phagocytosis will happen by the who is performing here? Who is doing phagocytosis? None other than phagocytes. Okay. Then, and I have already discussed this thing. How the phagocytes can easily identify the apoptotic cell. Okay. Then again you can just see.
So you know, from the extrinsic pathway which caspase enzyme got activated? 8. From the intrinsic pathway which caspase got activated? 9. So again, please initiate the caspase. These were initiator caspases. Okay. Caspases are divided into 2: initiator and executional caspases. Initiator caspases you know as 8, 9 and all. Executional 3, 7, 6 and all. So after all this pathway, it will go and activate rest caspase enzyme. I told you caspase means lot of enzymes are there. So it will go and activate some other caspases. So these were like executional caspases 3, 7, 6 and all. This executional caspase means execute the apoptosis. So it will undergo apoptosis, like the nuclear matrix will get broken, the cytosol damage will happen, nuclear matrix fragmentation will happen. All this degradation, all this organelle degradation will happen. Everything will happen, and then finally it will form the apoptotic bodies. And who will come for phagocytosis? Phagocyte. So this is the thing. And one more thing, how the phagocyte can identify the apoptotic bodies, isn't it? Even apoptotic bodies are also there. Our normal cell is also there, right? So normal cell is also there. And how phagocyte will find out these are the apoptotic bodies? I should go to the apoptotic bodies. I should engulf the apoptotic bodies. How? Because in our normal cell, this is like a normal cell. Okay, this is a normal cell. In our normal cell, there is something called a phosphodiesteracyl serine, which is also present inside the cell. I mean, the position is like inside. Okay, it is inside for a normal cell. But for apoptotic cell, for apoptotic bodies, this is present outside. This is present outside. So what labeling is there? So at that time, phagocyte can easily identify which is the normal cell or which is the apoptotic cell. Just you have to remind this thing. In normal cell, this thing, this phosphodiesteracyl serine, this will be inside. So the phagocyte will not go and bind, or it will not go and take or engulf the normal cell because it is inside. These are present inside. In apoptotic bodies, this will be present outside, so that it can easily identify, and it will only the apoptotic bodies will undergo the